Good evening, everyone. Assalamu alaikum. I'd like to welcome you all to this online webinar. This is a continuous medical education activity from King Khalid Eye Specialist Hospital and Research Center. And it's my great pleasure to welcome and introduce Professor Malik Kahook. He's a professor of ophthalmology in the Slater Family Endowed Chair in Ophthalmology at the University of Colorado School of Medicine. He is vice chair of translational research and serves as chief of the glaucoma service and co-director of Fellowship at the University of Colorado Eye Center. He was named the new inventor of the year for the University of Colorado in 2009 and the inventor of the year for 2010. Dr. Kahook also received the American Glaucoma Society Innovator Award 2020, AAO Achievement Award 2011, AAO Senior Achievement Award 2017, American Academy of Ophthalmology Secretariat Award in 2014. He was also named top 10 list of the Ophthalmologist Power 100 list in 2020. And he also serves as a member of board of directors at Orbis International, and he's the co-founder of Eyeglass. Professor Kahook, thank you so much for being with us today. It's an absolute pleasure.
Thank you for having me. I'm looking forward to giving the talk. I'll leave about 10-15 minutes towards the end of the first hour here to answer any questions. You can also put them into the chat and I can address them towards the end. The purpose really is to just give a general conversation, a general talk about translational research using examples from the University of Colorado. So I'll go ahead and get started. And as is traditional, just talk a little bit about my disclosures. I do work with some companies that probably most of you are familiar with, Alcon, New World Medical that makes the Ahmed valve, and have some consultancies with a couple of other companies. So just keep that in mind as I'm going through my slides. So I don't know how many of you have seen this movie, Free Solo, where this insane person climbs the side of a mountain without a rope. And I like to put this as the first video because oftentimes this is what translational research feels like. When you're first starting out with an idea, you sort of feel alone that you're climbing and trying to get to the peak, but you find out pretty quickly that you're not going to get there without a lot of help and a lot of people around you. The discussion that we're going to have today is around what innovation, where does innovation come from, steps to get started. So if you have an idea, what are some things that you can do to get started? I'll talk about 3 real-world examples that came from our laboratory here at the University of Colorado. Every time you see this emoji, I'll tell you a little bit about the mistakes that I've made along the way and that hopefully you won't repeat. And then at the end, I'll just talk about 5 tips for you to consider. When we talk about innovation, I think one very important point, especially for this audience, is that the majority of innovation that we use on a daily basis, whether it's in clinic or the operating room, comes from physicians. Phacoemulsification came from Charlie Kellman. All of the drainage devices came from MDs, in one case PhD with Dr. Mateen Ahmed, who made the Ahmed valve. OCT, the majority of those involved in the development of OCT were physicians, and all of them are still practicing in ophthalmology. Lasers came mostly, and when I say lasers here, I'm focusing more on the lasers that we're using for cataract surgery. Kurtz, Kruger, lots of physicians were involved in the early days. And then practically all of the MIGS devices that we're using to treat the angle have come from physicians, with Dr. Hill coming up with the eye stent. Barvelt, who did trabectome. Lots of innovation in the angle space. And again, almost all of them, if not all of them, came from physicians. But it's not just the big devices that we talk about with the big machines or the high profile items. We also have a lot of devices that we use on a daily basis that are just forceps. For example, for the Fechner forceps, the Akahashi pre-chopper, iris hooks that came from Jean Dewan, who's a retina specialist, and the Seibel chopper that I use routinely as my second instrument for cataract surgery came from Dr. Seibel. If you're thinking about ideas, if you have some ideas, I like to just put this slide in here and you can come back to it. You can take a screenshot and come back to it. If you're struggling with how to articulate your idea or how to solve something that you're seeing in the operating room and you don't quite have a solution, these are the 4 main ideation principles, and they center around what you see in the box here on the bottom. Innovative ideas often emerge by reimagining the possible. Drawing inspiration from the improbable, that's science fiction. So if you're watching science fiction and they have a communication device on their sleeve that they use to communicate with each other, A lot of people have taken that, and that's how cell phones came to be. They were trying to imagine what is the impossible and what comes from science fiction, and that's what led to it. Leveraging analogies. So in the US, there was a company called Circuit City that did a lot of electronics. Didn't work out so great for that company, but they took everything that they learned and they used it to sell cars. And that has been quite successful for them. You can also challenge assumptions, first principles. So thinking from the basics of things, just dismiss everything that we know and just start thinking fundamentally about the problem as if nobody has worked on it from day one. And that's similar to what SpaceX did for the reusable rockets, where in the past you would lose all of that material, but now they're being reused and it's making space travel less expensive. And then repurposing. This is an important concept that is used even in medical technology. So if you have technologies or if you have pathways to do one certain thing, like Amazon selling books, clothes, shoes, everything online, they needed the infrastructure to do that. And they repurposed that infrastructure to be one of the main cloud services in the industry, and that's AWS. It's also the concept behind feathers that were initially used for warmth, but then became highly assistive for flight. So again, these are just four ways to think about it. These are the four main ways to think about ideation. And it's good for you to go back to these principles when you're thinking about new ideas. I was trained by Joel Schumann, and Joel Schumann was trained by David Epstein. And David Epstein was very popular for going around and talking to people about how to come up with new ideas. And I met with him when I was in fellowship. He's since passed away, but had a lot of great ideas. And I put one of his lines here so you can come back to it and read it. He believes that the laboratory that we use as innovators comes from the clinic, comes from the patients. Patients tell us what's important to study and whether or not the treatments have any efficacy. So we have to listen to our living laboratory in the clinic and then be attentive and vigilant and never complacent when we're hearing things from patients. And quite honestly, that's how most of the stuff that I'm working on comes to fruition. It comes from patients in my clinic, or I'm in the operating room and I'm doing something and I think, there's probably a better way to do this. And that's usually the genesis of an idea. One of the big things that we have to ask ourselves, and this is probably one of the harder questions, is do I have a good idea? You might have an idea, but is it a good idea? New does not mean better. So if you have a new idea, that doesn't necessarily mean better. New does not mean practical. You might have some knowledge about a way to do something, but it might not actually be manufacturable. You might not be able to scale it up from a production standpoint. And new does not mean viable. It doesn't mean that the community of clinicians, ophthalmologists in our case, will accept it. So you really have to think through your idea and be very honest with yourself about whether it could translate into a commercial product that would be used, eventually reaching the patient. So we talked a little bit about where innovation comes from. Innovation comes from identifying an unmet need, saying out loud to yourself when you're just kind of contemplating, is there a better way, a new way to do this? Is there a safer way to achieve the goal? There might be some ways that are practical today, but just not as safe as they can be. And then the real question becomes, how do we get from idea to solution? This is a cartoon that I think really exemplifies that last statement. This is a beaver standing in front of one of the major dams in the United States, and the beaver's thinking, "You got to be kidding me! I'm the one who invented this." Now, invention oftentimes is the easy, simple part of it. You can cut an idea; it could even be a great idea. But the hardest part about doing that is making it workable, making it affordable, actually building the infrastructure to get that technology to the market. So don't think of the idea as really the end game. That's really just the start of any journey that you might be going on. So it all starts with an idea, as I said, and unfortunately, this is the easy part. The majority of inventions that we come up with are conceptual. It's a physician who's talking to another colleague. It's residents speaking amongst themselves about a new way to do things, but it oftentimes does not go beyond the conceptual to that next stage. And the major limitations include time constraints. We're all busy. We're all seeing patients. We're all focused on the things that we were supposed to be doing when we went to medical school and then went through training. And then we also have a lack of know-how. We don't go through the innovation process in medical school or in residency. We don't go through the patenting process. We really don't understand how to translate ideas. It's an additional skill set that we have to build along the way. There's also a lack of finances to push this forward. Um, we are risk averse in nature as physicians. We always want to do what's right. We want to do what's safe. Spending money on patenting and new devices, coming up with a proof of concept device, all of this is something that's against the nature of most physicians, and that is a huge barrier to getting the idea to move forward. So, how can we push forward? I'm going to talk about three different technologies that came from the university. The first one is what we classify as a class 3 device here in the United States. It's a more rigorous pathway to get a device from conception all the way through into commercialization. I'll talk, the second idea is an early stage novel material, just giving you a different idea of the innovation pathway if it's more focused on a material rather than the final solution. And then a class 1 surgical device, which is an easier pathway compared to the class 3 device. And for that, I'll focus on the KDB device. So let's talk about the class 3 device. This was a company called Clairvista Medical. This spun out of the university into an incubator over a decade ago. So it's been a long time since this technology spun out. The technology itself, you see on the left-hand side, it'll become a little bit more evident what it is when I play the animation next. But this is a two-piece intraocular lens. The blue part that you see is a base that is a donut. There's a hole in the middle. That goes first into the capsular bag, and that's designed to be very stable so that once it's in place, you know what the lens position is, and you don't have to guess what the effective lens position is going to be, which can help you pick the right intraocular lens power. It's also designed to not rotate. So if you put in a toric, it's very stable. And then if the optic goes in, and let's say it's a multifocal that the patient doesn't tolerate down the road, you can take that lens out and put a new lens in because it's exchangeable without touching the capsular bag. That's as opposed to a standard intraocular lens that you see on the right-hand side. So here's what the animation looks like to give you an idea. The base is already in the capsular bag. The optic then goes in just like you would inject any intraocular lens today. And then with a standard Sinskey hook, the lens is placed into the base, which allows for this optic to be exchanged without having to touch the capsular bag at all. So if there's any fibrosis or anything like that, it's not something that's going to impede removal of the optic and putting a new optic in. So the stability allows us to achieve better outcomes with primary surgery. We can better predict the power of the lens, and it does not rotate. And the modular design allows for an insurance policy to address the needs. Let's say in the future, 20 years from now, we develop a much better optic that everybody wants. Now you would have the option to do that and to be very flexible with how you're caring for each eye. The second technology is the material technology. This is to address the unmet need of intraocular lens materials that need a balance between an incision size that's very small, but yet having the mechanical ability to hold itself in place. We can make very, very soft lenses that we inject through very small incisions, but they tend not to keep their form in the capsular bag. And you can also do more rigid lenses that keep their form in the capsular bag better, but then they require a bigger incision. So our goal was to come up with a material that balanced those two things. And the solution for us was a shape memory polymer, a specific kind of material that we could either mold or lathe, the standard techniques for making intraocular lenses today, and still meet the two targets of small incision and great stability in the capsular bag. When we were working on this material, there was no other shape memory polymer that was being used for intraocular lenses. So this was going back to the fundamentals and saying, how can we replace the standard hydrophobic or hydrophilic acrylic materials that we use more commonly? So how do we set ourselves up for success? Breaking through from the conceptual, as I said, most of our ideas die in the conceptual phase, and then taking them into the commercial phase requires a significant amount of commitment with very big steps. The first step, as we just mentioned, is defining the unmet need, doing the market research. This can be a very boring process. It's not what physicians are typically doing on a daily basis. It's not in our skill set. I'm going to talk a little bit about prior art search and what that is, and some of the mistakes that we've made in the next slide. You have to talk about, can you build it? Can you manufacture it? We'll also talk about that a little bit. And then if all is a go, it's time to protect your idea, and we'll talk about that a little bit as well. So prior art is the term that we use to describe what knowledge is present when you're coming up with your idea. So if somebody comes to me in our department and they say, I have this new idea for something that's going to be great, the first thing that I ask them is, did anybody else do it? Has anybody else tried it in the past? Have you read textbooks, other patents, articles in our journals to make sure that it truly is novel, that it's something that nobody has done before? You should seek a patent attorney who's familiar with the space, and that would be very helpful to set the foundation. Talk to others who've been through the process of patent writing and discuss the mistakes. Were they too narrow? Were they too broad? Were there any freedom to operate issues? And then here's a mistake that a lot of physicians make. When you have an attorney that you're working with, there's an assumption that the attorney is going to do the majority of the patent writing and the molding of the patent. And that's not true. We are the subject experts. It is up to us to do the heavy lifting from a content standpoint and to make sure that as the attorney is helping you with shaping the patent, that you're really driving the process. If you don't do that, the output isn't going to be perfect. It's going to be something that's more on the legal side of things rather than the novelty side of things. Unless you're very lucky and you get a patent attorney who's very accustomed to the ophthalmic space and has a significant amount of medical knowledge. And that's not very common to find. So just keep in mind that you're going to be doing a lot of the heavy lifting. And then we keep coming back to the financial issue. What's the roadblock to getting a patent? It's not just a timeline process, it's the cost issue. But an application for an initial patent in the United States is around $3,000. So it's not that dissimilar in different parts of the world. In fact, it tends to be less expensive in different parts of the world outside of the US. So it's not a major block, but it's significant enough that you should be thinking about it when you're starting the process. When you're thinking about what you may be able to patent, I would say you should suspend your judgment about what you think is patentable or not patentable. I've seen many things that have received patents, have received claims that initially people thought could not be patentable. And I throw these examples, these three examples, so you get an idea of what has been patentable in history. You know, glasses for chickens on the left-hand side. In the middle, a mechanism to help somebody tip their hat without actually touching it. And then on the right-hand side, if you're on a flight or a train ride, you can actually put a hammock between two seats and have a little bit of rest rather than just sitting up straight for the whole trip. All of these things you might have looked at and say not patentable. You see exactly the same thing in the medical field where you can be narrow enough, specific enough in what you're thinking about doing, and still be able to achieve a patent protection. And this is a much more intricate topic. This could be several talks within, a series of discussions around innovation. But it's a very important part of what you need to do if you want to get into the innovation process and have devices on the market. So what do you do when you start thinking about building the proof of concept devices? We hear a lot about people doing this in their house. This is usually the technology companies like Apple or Microsoft. Early on they would work in a garage or somewhere in their house. That's not typically the case when it comes to medical manufacturing. We typically have to seek partners, and it's better to seek partners who are local, somebody where you can drive over, walk over, and meet with them, have regular touch points to discuss exactly what they need to do, how they're doing it, are they meeting their timelines, and to make sure also that the amount of spend initially is reasonable. You don't want the cost to get out of control and then have that dictate how quickly you can move early on. Proof of concept devices can be very simple, with the point being to learn and fail fast. You don't have to make a device that is completely final from the first go. You really just want to get an idea. Is there a pathway to get this refined, to get this to be much better? So don't think about perfection. Think really about understanding and how to get better for the next stages. You should also seek larger partners who can scale up production. This is a mistake that I've made in the past, where if you're working with somebody and they're small, they're not able to scale up. Then when it comes time to scaling up, they might transfer that information to a bigger partner who then can't do what the smaller partner was doing. They can scale up, but they can't do some of the intricate work that the smaller group was doing. So when you're first starting out with some of this work, you should find out if your partner can scale up and make thousands of these, and can they put in the processes that are needed to be commercial. Something you should be thinking about very, very early in the process. Sources of funding. You can see there's a theme in this talk talking a lot about the finances because it often does dictate how fast you can move and how far you can take an idea. These are the various ways for funding your ideas. The first one is self-funded. The good side of this is that there's no dilution. You own everything that you're doing. There's no headache dealing with partners or people who might have unrealistic expectations. The problem with this is that it's easier said than done, and it's really hard to know when to stop spending your own money. If you're spending your own money on ideas, there's really nobody looking over your shoulder to say, maybe it's not working, you should stop. So there's a tendency to overspend when it's something that's your baby, it's your idea, and you want to keep pushing it forward. Another idea is to do friends and family. You get more money this way because you're pooling everybody's money and they can potentially put in more money. But I would not recommend doing business with friends and family. So I think that's something that I've stayed away from. And I think you're getting back into that headache idea, which is a pro for the self-funding. So a lot of people choose to do angel investors. And there are professional angel investors who put in part of their money on a regular basis into small companies. They have deeper pockets. And a lot of times they can give good advice, but sometimes they have no knowledge when it comes to medical devices. They're just interested either in you as the inventor or the idea in general, but really can't help you along the way. The downside of this is that there's going to be dilution. If somebody's putting in money as an angel investor, they're going to expect that they own a piece of that technology when it comes to market. And I think from my experience, the expectations can be unrealistic for angel investors. They don't necessarily understand the entire process of what's needed, how long it's going to take. Now, of course, there are exceptions. There are some angel investors who can do this better than most venture capital groups. But in general, the angel investors are more hands-off, especially in the medical device world, and they have unrealistic expectations for how fast you can go. In an academic setting, there's university funding, typically no dilution, and there's a greater chance of success in getting the funding from the university. There's not a lot going on from the competition standpoint. It's just a smaller pool of competitors going after the money. But on the downside, when you're doing it at a university, you're typically on your own. You don't have a lot of resources. You're not hiring a bunch of people into a new company that can help you push the idea forward. And there's a significant amount of hurdles and red tape on campus, things that prevent you from going faster. So university funding does have a lot of upside to it, but the downsides are real, which is why you see a lot of technology after initial work at a university usually spins out into a new company, a smaller company that can hire people in and push faster. There are government grants, which in the US we call them SBIR grants. There's a high rate of success for getting those, but it is cumbersome. It takes a long time to get the paperwork done, and the money is usually not enough to get you very far in the innovation process. So that leaves most of us with VC funding, and you see that's the bottom right-hand side, VC seed rounds, A, B, C rounds, and so on. On the pro side, these are serious partners who know the field that you're in. They can be great advisors and help you along the way. It also gives you validation. So if somebody who has a lot of knowledge is putting in a significant amount of money, they're saying this idea is good. We bet on this idea, which can then get a lot of attention from people you want to hire and maybe bigger companies who might want to buy your smaller company one day. The downside is the same downside anytime you bring in outside money, which is the dilution. There's a significant amount of dilution, loss of shares of the company as you take more money with each round of fundraising. So busy slide that gives you a good idea of what the different routes are for getting money. But this is often central after you have your initial work done to figure out how you're going to pay for all the work that's needed. And then you can pick the one that best applies to your goals. So let's go back to ClairVista. We talked about the modular lens. We ended up getting venture funding for this company. We went through multiple design iterations, 104 designs, 8 device builds to figure out exactly which device we were going to take into clinical practice. Now, remember, this was a freestanding company without its own materials, so we had to go through our own material selection and testing. We went through 20 different materials and brought it down to one that we took into the clinic. We had to do our own bench stability testing to show that the base was as stable as I said it would be. Nobody had done any type of testing like this, so we had to design our own testing setup. We then went through animal model testing and biocompatibility. We went through injector optimization. There's no injector designed for a base, so we had to do some work and eventually found a partner that could help us with the injector. We visited with a lot of key opinion leaders in ophthalmology. We did a lot of testing and got a lot of advice from them. We also got regulatory help to make sure that we could get it through the FDA here in the US and then abroad. And then we did a plan for a proof of concept in humans. All of this was done before setting foot into the operating room, and it took about a year and a half to get through, which is actually pretty quick to get through all of that work within a year and a half. We had some great partners, some great consultants that helped us along the way. But it wasn't as simple as just saying, I want to do a modular lens and then getting that done in a couple of months. We had to spend a significant amount of money over a significant amount of time, and then eventually got to the device that we could take into patients. Shape Tech for the material, instead of spinning this out and taking venture money, we used university funding and grants. This was a partnership between ophthalmology and bioengineering. We iterated hundreds of formulations of the shape memory polymer, and we were looking to mix and match to reach the right balance of optical quality, stability, injectability is a huge one because we don't like very big incisions in ophthalmology. And then how cheaply can we make this, right? So how can we actually save as much as possible and make the final build as inexpensive as possible so that the cost of goods, the final cost of goods was something that could be commercializable. We did injection testing and injector innovation, similar to what we went through with ClarVista. We then did a lot of work around manufacturing. Could this be scaled up? We did our own animal studies, and then we prepped for the FDA studies as the next step. So, from a regulatory standpoint, you've heard me mention this a few times. A major factor in what we do, not just in the US, but internationally, is to get through the regulatory process, which can be quite rigorous. For us, it's the FDA, similar setups in most countries around the world. It's really difficult for physicians to learn the setup, and it's different in different countries as far as what the different time points, what's needed to satisfy things along the way. The FDA website in the US can be a resource, but it's really hard to navigate. You can talk to industry colleagues about their experiences, and oftentimes I find that they're very willing to help. And then seeking FDA consultants is extremely important. This is one of the things that we didn't do well early on. So now what I do is I seek FDA regulatory consultants very early in the process, and the money that's spent on that is very well spent to make sure that you're doing everything early on in a way that's going to set you up for success. It's just not something that physicians are usually trained for. So, perfect time to take a sip of my coffee as you're watching this about focusing. One of the issues that happens when you're in the middle of all of this is it's really hard to focus. You start seeing squirrels and things running around in the trees, and you want to focus on those, and you don't really want to focus on what you're doing. And that happened a lot with most of the technologies early in my career. I can tell you we got very busy with ClarVista. There were a couple of years spent just traveling to different countries that were doing the early implantations. And you can see the different sites around the world. This is literally all around the world from, one side to the other. So staying focused during that time, making sure you're getting the right data, making sure that your team is coming along and understanding everything that you're doing is extremely important. We completed the proof of concept studies for Shape Tech. We did all of the standards that were required. We kept things mostly in Colorado, so we didn't have to travel a lot for Shape Tech as opposed to the previous slide for ClarVista. And we spun off the company off into an off-campus facility, which was literally just right next to the campus, so very convenient to walk to and do the work. We started meeting with the strategic companies, with the Alcons and J&Js of the world, and we started working with contract manufacturers. We also met with key opinion leaders and had them play with the intraocular lenses that we made to inject them to go through small incisions in cadaver eyes to see exactly how it works. And we were off and running with ShapeTech. Now the big question becomes, what do you do to partner? What are the different pathways? The most popular pathway that you hear about that gets a lot of attention is the strategic company collaborations, whether they buy the company or they partner with you. If a strategic company is interested and you see many of them on the right-hand side there, it gives you affirmation that your idea is a good idea. Companies that are bigger are slower to execute. So the problem is if you partner with a bigger company and they have significant control, which they usually do, then you're not going as fast. And oftentimes, if they lose interest, then it's not going to make it to market. And that could be a huge setback for the technology. I would recommend this only when the idea is mature, when you've built enough of a body of knowledge around it to show the bigger companies that this has commercial value. Otherwise, there's a higher risk for them not pushing it forward. Venture or other investors, similar to what I said above, but they remain engaged and they can help build value. And of course, the third option is just go it alone. So if you have the finances to do it, you can do the build alone, you can do the testing alone, you can hire consultants to help you. It's certainly one option that you could do. So with Clairvista Medical, the decision was to sell. We ended up selling to Alcon prior to doing any of the phase 3 studies because the phase 3 studies were going to be quite expensive. We were only 5 years in when the company sold, which is a very short amount of time for an intraocular lens. So we thought that was good both for the members of the company as well as the investors. Alcon has the biggest commercial footprint, so it was good from a financial standpoint for the company, for the university. And also because they have many different optic technologies, we thought it would be a good fit with Clairvista. There was a family of next generation IOLs coming from Alcon at the time when this acquisition happened. PanOptix was just starting to be discussed to come into the market. And the price was right. Like I said, it just felt right for everybody involved. So that was one pathway that we took with Clairvista. For ShapeTech, we took a different route. The decision was to partner with J&J. When we initially partnered with them, it was AMO. This is prior to AMO getting acquired into Johnson & Johnson. This was a milestone-driven joint development agreement. So this was a big deal for us. We were able to use the resources within J&J to learn from them, which was hugely important, still important to this day with the stuff that I'm working on. And we were able to use their engineers and their manufacturing resources. So it gave us access to knowledge base and built-in customer because we were working with J&J. It also allowed us to increase our bandwidth and to expand into other work that we were doing. For me in particular, it allowed me to start focusing on some of the other technologies that we were spinning out of the laboratory. Learning from the inside was definitely worth the deal in itself. So there's a third option, the third technology, and I'll spend a little bit more time on this. I think many of you are probably familiar with excisional goniotomy. All of the development for KDB was done in-house prior to partnering with New World Medical. We iterated on the design, which I'll talk about here in a little bit. We got to proof of concept. All of this before licensing the technology to New World Medical. So on the pro side, we had full control. We built the value as much as we could, and we went on at our own pace, so I could run as fast as I wanted to. On the downside, we didn't have a lot of resources in the university to help do this, so I had to do a lot of it on my own. There is a significant amount of barriers or red tape to doing this. And then there's also uncertainty. You can push forward, but you don't have a partner, so you don't know at the end of that road, are you going to have somebody to commercialize your device? So excisional goniotomy became a big part of what I was doing here at the university. It took up a lot of time. When we talk about goniotomy, most of us talk about Barkan in the 1930s, working on it for pediatric and showing that it didn't work great in adults. But goniotomy went back many, many decades before Barkan. And then you can see the timeline since Barkan with Quintana, Dominguez, eventually gonio-curettage and trabectome, all before coming to the more modern devices and modern techniques like GAT and KDB. So there's a huge track record here that we were following and learning from along the way. The genesis of the idea for KDB is something that I like to talk about just for anybody who's listening who might have, your own thoughts and own ideas about what you might want to do. When we started KDB, it had actually nothing to do with surgery. We weren't thinking about building a surgical device. I was trying to figure out a way to get trabecular meshwork strips for some of the imaging studies that we were doing. So we were doing a lot of two photon microscopy at the time. And the two photon microscopy necessitated getting strips of trabecular meshwork in a reproducible way. And if you've worked with trabecular meshwork, it acts like wet tissue paper. If you're trying to peel it, it just kind of turns into pieces. So I went into the lab and I tried to make devices that would allow me to make a strip of trabecular meshwork. We went through multiple iterations. And the final design worked really well in the lab, where everybody in the lab was very happy that we were able to get trabecular meshwork strips. And all of this was done before deciding, hey, wait, this might actually be a good thing for the operating room. a lot of the work that was done on the ramp part of the KDB device. We found out very early on if you did not lift the trabecular meshwork and put it on stretch prior to making your dual incisions, that oftentimes it just crumbled, bunched up, broke up into pieces. So then it became a matter of figuring out the exact dimensions for the ramp. And we worked in multiple iterations to get the right lifting. And all of the lifting happens before the two incisions, the two blades are actually engaging the trabecular meshwork. So it took quite a bit of time, but we got to that point in a way that we felt was reproducible. And before partnering with New World Medical, which gave us a lot of excitement about what it could potentially do on the market. These are just the steps. So the tip was designed so that it would enter into the canal easily. Then after that, as I just mentioned, the ramp would engage with the trabecular meshwork and lift it up, and then the dual incisions would be produced to give you that regular reproducible strip of trabecular meshwork. We did the initial studies to compare versus other methods, including MVR. So on the left-hand side, you see with an MVR blade, you get more of an incision of the trabecular meshwork. The arrows are pointing towards the residual trabecular meshwork, which is significant, and those can reanastomose, they can close. And then that arrowhead that you see is pointing towards scleral injury, a cut in the scleral wall. And that is exactly what you don't want to happen because it can lead to inflammation. Now, on the right-hand side, you see what KDB can do. It removes the majority of the trabecular meshwork. The arrows are pointing at the TM remnants, which are not leaflets within themselves. They're basically just the residual tissue adherent to the wall. And then you see an open Schlemm's canal and no injury to the sclera. And we did this reproducibly versus other devices. You can see this is way back in 2013. So it's been 12 years since we did this work, just to show that there is a different way for doing goniotomy. And it turns out in clinical practice, this is extremely important, and why we used to say that goniotomy doesn't work in adults. It just turns out that incisional goniotomy doesn't work very well in adults, but excisional goniotomy works remarkably well in adults. This is one of the first cases. This is my friend John Berdal doing his first case. And you can see he just goes in, goes left to right, and maintaining a pretty good view on gonioscopy, which is important with all MIGS procedures, and then going right to left until you meet up with that area where you stopped on that initial sweep. TM acts very much like Velcro onto the iris. So you'll see some of that TM was on the device, and then there's just another small strip of TM that he goes in and he grabs with his duet forceps. You'll see the whole strip come out there. I'll do one more video with Paul Singh doing his first case. He takes a bit of a longer sweep from right to left and then reverses course left to right. In this case, you'll see the entire trabecular meshwork strip adherent to the device, and it comes out with the device. You'll see it sort of flapping in the aqueous right there. And then the device comes out, and you see the anterior wall of the canal white, showing that you've removed the trabecular meshwork. Now, as glaucoma specialists, we like our data. Whenever we see something that's new, we first thing we ask is, tell us about the data. So we started publishing papers on this when it came out into market. This is one of our papers from 2018. And you can see no matter where the starting pressure was, 29, 23, 18, these patients went down to the mid-teens, which was really good to see that you can get them down significantly low. Even if you started out at a lower pressure like 18, you could still get a few millimeters of mercury lowering. We also looked at longer term follow-up with excisional goniotomy that was accompanied with cataract extraction. The take-home message here is similar, that no matter where the starting pressure was, even if it was as low as 13, you're still getting all of these patients moving to a pressure that's somewhere around 12 to 13. And the patients who had a lower starting intraocular pressure, the majority of the time, 85% of the time, were able to decrease at least one of their medications, which is a big goal for patients that start off at a lower pressure. We did multiple other studies that had similar findings. This is excisional goniotomy with or without cataract surgery done just at the University of Colorado. Very similar results, getting out to the low teens with a significant reduction in medication. We also started doing studies in patient populations outside of the mild to moderate primary open-angle glaucoma, which is the traditional patient for MIGS devices. In this case, this is Cyril Duraraj, who partnered with some colleagues in Vietnam and published on angle closure glaucoma. And you can see that it was a dramatic decrease in pressure from 25 down to 13. from a mean standpoint. And this is reproducible across most of the patients that we treat with excisional goniotomy. And this is our standard of care now. And I think it's standard of care in many places around the world where if you're faced with a chronic angle closure patient, you're doing phaco, combine it with a goniotomy, these patients end up doing very, very well. We also looked at other types of glaucoma, like uveitic glaucoma. My partner, Mina Pancheva, has clinics that are dedicated to uveitis and glaucoma. She did a bunch of patients, and you can see from an IOP lowering standpoint, these very tough glaucoma patients can still get a significant amount of pressure lowering that's sustained for an extended period of time. We're still following these patients, and these patients are doing very well. And then from a study standpoint, I'll close off with this. One of the big things when you're going through innovating devices, it's one thing to do studies and to publish them, but I think the gold standard is to do a head-to-head RCT to compare to something that's already there to show that there might be some benefit. And that's certainly one of the things that was missing from the MIG space. We don't have a lot of head-to-head studies in the MIG space. So here what we did is we did a head-to-head study, phaco iStent versus phaco KDB. And what we found was a significant number of phaco eyes, significant compared to the iStent eyes, met the primary outcome of IOP reduction greater than or equal to 20%, or IOP medication reduction greater than or equal to one medication. So this was a really nice way to show that without leaving a device behind, we were able to use one of our inventions to get very low pressure with reduction of medication. And doing it with an RCT with a head-to-head gives a lot of leverage when you're talking to users to say, you don't just have to trust what I'm saying, you can see the data and see how it works. We still have much to learn. Now, one of the exciting things about doing goniotomy is we took it from the laboratory into the patient, but now we're going from the patient back into the laboratory where we're harvesting tissue and we're looking at it under the microscope and doing various studies. We're looking at samples. This is a sample that you see on the right-hand side that was sent over to me by Devinder Grover and Ron Fellman. They had done a GAT on this patient. The TM had scarred over. They went in, did a KDB, and sent me the results. And you can see, if you're familiar with histology, what you're seeing on the right-hand side is a significant amount of scar tissue around the trabecular meshwork. So we're starting to learn about the scarring in the angle after we do different procedures and potentially how we can overcome that with different medications or different designs for what we're doing from a surgical standpoint. This is a patient where I went in. I'm going to stop it here just for a second and reverse just so I can set it up for you. So I'd seen this patient in the clinic, and I had done an iStent a couple of years prior. And I noticed that the iStent was actually moving back and down. So it was not in its initial space. So over the last few months, it was just kind of being displaced. Couldn't really tell why, but the pressure was going up. So we decided to take this patient back into the operating room. The iStent came out very easily. You can see I didn't have to struggle at all to pull it out. And then I go in with KDB, and I could tell right from the beginning that the experience was very different than my typical KDB. I was actually feeling some resistance as I was going through the canal. And I didn't really appreciate why until I did my left to right sweep. And I'm going to zoom in now, and you're going to see that there was a film that was developing over the trabecular meshwork that was vascular and fibrotic. And that was right over the area where the iStent was sitting. Here it is. So I collected this tissue along with the trabecular meshwork and sent it in for analysis with some of our laboratory colleagues. And what we found was similar to what I showed in that first slide from the post-GAT patient, that there was a significant amount of fibrosis, which you're seeing on the left-hand side. And we are seeing this with every TM that we're harvesting around iStents. So when our iStents are losing their control, which of course happens in every glaucoma surgery eventually, we're taking the iStents out, we're collecting the trabecular meshwork. We published this paper in 2018 to show that it's reproducible. The patients who have either an iStent or a Hydrus, they develop the scar tissue just like you do around an Ahmed valve or a Barvelt where they encapsulate. The same exact thing is happening with the devices that we're leaving in the angle. And this is going to help us understand how to overcome some of these barriers in the future. So the previous dogma that goniotomy does not work in adults, I think, has been wholly disproven. And that's part of the fun part of doing the research and the innovation, that we can do things that are new that people didn't expect in the past. I won't go over this in too much detail, but it's just to say that we don't just face obstacles in the medical field with new inventions. It's really anytime you're trying to change the status quo. When cars first came out, people with horse carriages were very upset. when the Horse Association of America for many years fought against the tractor. They didn't want tractors being involved in farming. So it really does take a little bit of time before people buy into the new ideas potentially being better. Resistance often stems from the fear of disruption rather than genuine concern. It's just we're creatures of habit. We like what we like, especially glaucoma specialists, especially ophthalmologists. And adoption of innovation, with few exceptions, I think does benefit society once those initial barriers are overcome. Just takes a little bit of time. One example of that is Dr. Ahmed. I always like to speak about him a little bit. And I met him after training and started working with the Ahmed family at New World Medical in 2013 or so. And when I learned more about his history, I thought it was interesting enough to share that. So he's a biomedical engineer by training. He didn't start New World Medical until he was 45 years old. So it's not the traditional 20-year-old starting a startup company. And through strong belief in serving humanity, which is the main drive for Dr. Ahmed, he created the AGV, traveled the world. He himself trained physicians on how to use the device. And he was able to kind of push through that fear because before the idea of having a valve device was just not something that caught on. So he was the first to push that through. And now around the world, standard of care is having the AGV in your operating room whenever you need it. Just briefly, the innovation triad, we're not doing the stuff alone. Just like I started off with the free solo climb on the mountain, we have many people helping us, technical experts, engineers, business experts talking about the business and how to do things. And then, of course, that's where we come in as clinician experts to say what may or may not work in the eye. But it does take all of this triad in order to advance the technology. I'm going to just finish off with a few slides talking about the fun part of what we do from an innovation standpoint. You see a lot of people here that I've worked with in the past. This is the Clairvista team after the acquisition, just having a get together. We still work together, a bunch of us do. Chris Hafner is in our current company, Spyglass. Rudy works in Spyglass as well. Glenn was the co-founder of Spyglass, and Paul Yu still consults with us from time to time. So the team kind of stays together. This is the New World Medical team. This is Bilal Khan, who's CEO of New World. This is Khalid Mansour, who is the Chief Operating Officer and President of New World Medical. Just showing some good times here in Japan, just visiting some friends and talking about different technologies. This is Raphael Chan, who works for Alcon. And then here's my chairman, who's very supportive, Naresh Mandava, who's very supportive of the different innovation tasks that we do within the department, the different companies that spin out. I always like to show this photo just to show how happy he is with with a lot of the stuff that we do. And as long as he's happy, then I'm happy. We have done a lot here at the University of Colorado, just recognizing some of the colleagues we have in the department. Over 300 filed patents in the past 10 to 15 years. Over 100 granted patents. We've had 12 startups come through the department with 4 inventors of the year out of our department. We're a small department on campus, but yeah, we have 4 inventors of the year just in the past 15 years or so. Over 200 million in outside investment, whether it's VC or big strategic companies with multiple commercialized devices. We've treated hundreds of thousands of patients, and I feel like we're just getting started. So we have a lot of work to do. I've had the great fortune of working with different companies and different devices like KDB, Harmony with Clairvista, Spyglass, which is the current company that we have launched that is going into phase 3 trials in the United States and globally. And then I've worked on devices with New World, just helping out with their engineers with something called Streamlined Surgical System and other devices that have come out. So I've done things on different levels, whether it's my own device spinning out like KDB or starting companies like Clairvista or Spyglass, or just working with different partners on helping them with some of what they're doing. It's been a great privilege and an exciting part of my own career. Finishing off with 5 tips. And I'll mention these because I think if you can take screen grabs or just look at the talk later, these are things that really drive why you're doing what you're doing. You want to navigate your reality, academic versus private. Is it something that's going to be fruitful for you? Is it going to add to your fund of knowledge and how you think of your career? You always have to think about PAN versus non-PAN prior art search. Like I said, consider the regulatory process and how that might work into your day-to-day life. You have to build the right team. These are people that you're going to spend a long time with. Traditionally, it takes about 6 to 8 years to get a technology across the finish line. If it's a drug, it can take 10 to 20 years. So you're going to be working with the same people for a very long time. Make sure you pick the right people to work with. And then remember the innovation triad. It's not just you doing it. You have to have engineers, business-minded people. You're going to be working with patent attorneys, with different consultants. The physician brings a very important part to this, but there's no way that a single physician is going to do everything on their own and have all the knowledge to do it. And then along the way, just make sure you have fun. If this process isn't fun, it's not worth doing. You're not going to be able to get through all of the hardships that come up. So just remember to take a little bit of time to breathe and have fun. So I'll stop there. Thank you very much, and I'm happy to take any questions.
Thank you so much, Professor Kahuk. It's a brilliant presentation, a lot of lessons learned over the years. Um, I'd like to ask about, before we dive into questions from the audience, I was just wondering, uh, how frequent do you still use the Kahoot dual blade yourself in your clinical practice for both angle closure and open angle glaucoma patients?
So we have 9 glaucoma specialists here, and every single one of them actually did fellowship here. So they all had exposure to KDB during their fellowship. And we're primarily a non-implant group. So we don't do a lot of stents. We do a lot of KDB, Streamline, more recently Via 360 that just got launched from New World. And that's our mentality. Our mentality is if you can do an excisional goniotomy and not leave anything behind, then that's our choice. If you want to do something that's a softer touch, then we use Streamline. And so we're doing, as a group, we're still doing quite a few here. It's our go-to. We still do stents. We still do eye stents and Hydrus implants. We have a very busy fellowship, so we want the fellows to train on everything, of course. But I would say primarily we use things like KDB and Streamline here.
Okay, thank you very much. Going back to the your innovation journey, how what would you say the job of a surgeon innovator be? Stuck within the lab, developing drugs and devices, and then licensing it to companies the way you did with New World Medical, or do you think that you should take it a step further the way you did with Spyglass Pharma and then found a company, become an entrepreneur, and go through all those hassles as well?
So I think, look, it's personality dependent and it's resource dependent. I started off doing a lot more work in the laboratory and then found out that I wanted the control of having a startup company, that I wanted to be involved not just at the beginning and then hand off something. I wanted to be involved all the way through the phase 3 trials, through the regulatory approval. And I was fortunate enough to meet venture capitalists who were willing to put in the funding beyond what the university was doing. So those two things have to meet. It has to be your personality along with the capability, having the funding in place to allow you to do it. Now, I know a lot of people who want to innovate in the lab. They want to see their patients. They want to publish their papers. They don't want to ever leave academia, and they do just fine. And there's nothing wrong with that. It really just depends on your personality and what drive you have to do one thing or the other.
Great, thank you. I think we have a couple of questions from the attendees. Maybe we can address
Yeah,
those.
I see here. I can read through a couple of them. Let's just
Sure.
see.
This is a surgical question asking about bent ab internal needle goniectomy and how it
Okay.
compares to
Don't
Kahoot
see
goniotomy.
that, but I can answer that question. Yeah, so that's a common question that comes up when we're talking about goniotomy. And it's not just that. So GAT 360 essentially is a 360 goniotomy. In the US, we have devices like Scion, Trabex, Trabectome, which came before. All of these devices have pluses and minuses. The pluses for KDB I've already mentioned. It's really the reproducible excision without the collateral tissue damage. With bent needles, we did a bunch of those actually in the past, even before KDB came to the market, because we were trying to see if it was reproducible. In our hands and in the hands of most of the people that I've talked to, it's not as reproducible, and you're more likely to get injury to collateral tissue through a needle that's not designed for that purpose. So, from a results standpoint, I've seen papers that have published both, where initially, maybe for the first 6 to 12 months, there's about similar efficacy, but then after that, that's when the scarring and the reapproximation happens. What I always like to ask people who are considering the devices is, what would you do in your own eye? So if you had all of these things available, would you want a tool that was designed specifically for that? Or would you want to bend a needle and do it yourself? And what would you do if it was your own mother? So, and I think that's the best answer to the question.
I wouldn't mind the KDB if I had glaucoma,
Yeah,
but I would
yeah.
be afraid of the post-op hypema. Maybe that's the concern.
So this is a really important point, actually. I didn't see it as part of a question, but it's something we should talk about. There are pluses and minuses to everything that we do. And if you're doing GAT, there's a significant amount of hyphema. So we know that about 40% of patients post-GAT have hyphema. With KDB, it's anywhere between 10 and 25%, and it's usually a millimeter, so it's not as extensive as a GAT, but you can certainly get it. And that to me is technique related. There are some things we can do from a technique standpoint, like leaving a little bit of viscoelastic in the anterior chamber. If you just do a 10% fill right at the goniotomy site, we don't see hyphemas when we do that. So it's technique related. But anytime you're disrupting the tissue and disrupting the TM, whether it's a stent or a goniotomy or a canaloplasty, we have to know how to limit the amount of complications, but also how to deal with them. It's not just a straight phaco. It's a completely different way of practicing.
Yeah, thank you for mentioning that. We have a question about a person. I think we've touched on parts of that during the presentation, but maybe revisit that topic. Someone that has an idea for a surgical instrument, which is a very common scenario among surgeons. How would you think? How would you suggest they approach?
So somebody who has an idea for a surgical device, what do they do next? Is
Yeah.
that, yeah.
Yes.
So, yeah, it depends a little bit on where you are and who you have around you. So, if you are at an academic institution, oftentimes there are engineering groups that can help you. Oftentimes there are tech transfer offices that can help you with the patenting process. If you are not, if you're in a private practice or you don't have a university or facility surrounding you, then oftentimes you have to learn from friends, call people. I get a lot of messages on LinkedIn or by email asking me, can you introduce me to this person or that person? I think in our world, there's a lot of willingness to do that. And I like that's one of the things that I love about ophthalmology. A lot of people are willing to help, to the next step. I would say the number one predictor of whether you're going to do something along that continuum is how much of an internal drive do you have to do that next step. It's really not about how do I do that next step, is do you want to do that next step? So if you watch some of the YouTube lectures that we've given, or, I've done a few of these where I talk about the different steps, you can just get a piece of paper out and say, okay, I have an idea. The next thing is proof of concept. Depending on your idea, who do I need to call to help make this? Do I have university resources? Okay, I have proof of concept. Now the next step is to go do a provisional application or a patent of some sort. And then after that, it's I need money to go do more. I need team members. I need engineers. And then you start sort of getting in the mix of finding out who to call and how to learn from them. I can tell you when I travel, and I do travel quite a bit for work, I try with each trip to meet somebody that teaches me something that I didn't know before. So I'll ask them about something. I'll read their papers. I'll read about their innovation process, and then I'll try and get one piece of information that I can add to my list, and I do that with everybody, and that builds up over time. So I know who to call if something comes up in a space that I'm not as familiar with, or I learn something about a mistake that they made that I don't repeat. So I think watching as much as you can online, attending these lectures, and then keeping a list of what your specific idea is and what corollaries are out there to help you get to the endpoint that you're looking for.
Great and a comprehensive answer. Thank you. I think one blunt question asking about whether or not innovating is financially rewarding, or is it just a matter of luck?
So I can tell you that so I'm a university employee. I work for the state of Colorado, and everything that we do essentially comes through the university. The university takes good care of the physicians that work for the university, so we all do fine. And I don't think if you're doing it for the money part of it, you can probably do much better just doing a bunch of cataracts and premium IOLs and go into LASIK and do that stuff, right? But the rewards of knowing that KDB is being used around the world, that it's treated hundreds of thousands of patients, I think is much more rewarding than the financial part of it.
Yeah, I'm not sure if going to make you feel any different, but I personally, as a glaucoma specialist practicing in Saudi Arabia, KDB now ranks number one amongst the top MIGS procedure that I personally perform. So On behalf of all the Saudi glaucoma patients, just wanted to say thank you.
Yeah, I'll say shukran to that. Yeah.
We're at the end. I just want to wrap up with one final question or comment on a topic that I'd love to hear your remarks on, which is, you believe that innovators are born or they can be made? Is innovation something that, and I don't want a textbook answer, I want you to answer me from your personal experience. Growing up, were you that kind of person that loved to think about problems in a different way and had that inner drive to create solutions? Or is innovation a habit and a practice that we can teach and a culture that we can disseminate within any institution?
So it's a really hard question to answer, but to answer from the heart, as you mentioned, the innovation process you can teach to anybody. That's not difficult at all. What you can't teach, at least I'm finding this out, this is my personal opinion, is the willingness to keep getting up when you fall down. And when you're innovating something, I can give several lectures on everything that I've done that has failed. We've had a lot of devices that have failed, and you know about the stuff that makes it to the market, but we've had plenty of stuff not make it to the market. But there's always this willingness to say, I'm going to keep trying. I'm going to get up. I'm going to put one foot in front of the other. Not going to give up. That's something that's more inherent into some people and not others. And I think we see that in our trainees. We see it in our colleagues. So I think you can always teach the process, but you can't always push somebody to go through the process. And a lot of those people, I think, are born.
Yeah, thank you so much, Professor Koik. I think this brings us to the end of our webinar. I've attended previous webinars, and I don't think that we usually reach more than 100 attendees. So this speaks to how interesting the topic is, and that we are really fortunate to have a speaker like you. Although some of the attendees, I think, were AI agents taking notes, not actual human beings. So we were joined by robots.
That's great.
Thank you so much. Jazakallah khair. And I hope to meet you in person one day.
Inshallah.
Inshallah. Thank you very
Inshallah.
much.
All right. Thank you very much. Yeah. Wa alaikum salam.